Welcome, everybody. Why don't we just give 1 or 2 minutes for people to join, and then we'll go ahead and get started. Great. Well, we're already a minute after, so, and I see people rapidly joining. So why don't we go ahead and get started? Hi, everybody. I'm Marcy Lidback. Really appreciate you joining us today for, this is the 4th webinar that we've offered as part of the Year of Water through the AmeriFlux Management Project. And if you haven't been able to catch the other ones, they are on, they've been recorded and you can access them from the AmeriFlux webpage, so please do that. Today's topic is drylands. And what we're going to do is follow a format. We have 4 exciting speakers. They are each going to give a talk, and then we'll do Q&A at the end. So one exception to that is our first speaker, Dr. Steve Kannenberg, has to leave early. So please go ahead and put your questions in the chat, and he'll try to answer them before he has to take off. So why don't we go ahead and get started? As I mentioned, our first speaker is Dr. Steve Kannenberg. I put University of Utah in the announcement, but he has just recently moved to Colorado State University to do a postdoc there. So Steve, go ahead and take it away. Thanks very much.
All right, thanks a lot. Let me share things here. Okay, Marcy, full screen version, are we good? Okay, nice. Well, awesome. Yeah, thanks to you all for being here. It's nice to see this great turnout. Really excited to see all the other speakers in the session too, perhaps later when it's posted on the YouTube channel. And as Marcy mentioned, if you have a question for me, since I have to head out, just throw it in the chat box and I'll take a couple minutes at the end here to talk about it. So I want to start off by thinking about this picture here a little bit. This is a Pinyon Juniper woodland in southeastern Utah. I know for a fact that a good amount of you here are very familiar with Pinyon Juniper woodlands, but for those of you who aren't, I think there are some really kind of intuitive conclusions you can draw about this ecosystem just from this picture, which is, it's probably quite hot. It's really arid. The vegetation is very sparse. And it's for those reasons that I think, many people or many ecosystem ecologists, when first trying to think about this ecosystem, their intuition might be that, it can't possibly play much of a role in carbon and water cycling broadly. Well, that's not quite true. Arid and semi-arid ecosystems, kind of collectively drylands, really exert kind of a disproportionate influence on carbon and water cycling globally. And the reason for this is that these dryland fluxes are extremely variable across space and across time, and these systems can really rapidly fluctuate between a source and a sink of carbon. And it's this variability that makes dryland so important for determining the strength of the land carbon sink globally each and every year. So here's an example of just how variable some of these ecosystem fluxes are in this area. This is a graph courtesy of Mallory Barnes of kind of southern Arizona, southern New Mexico, gross per area productivity. And you can see when you're looking at some of these fine resolution data, just how heterogeneous GPP is across space. And you can't really capture it with a larger kind of resolution. You can also see just how, here's another example of just how kind of temporally variable fluxes of carbon and water are in this ecosystem. These are a time series of net ecosystem exchange and evapotranspiration, actually from the site in my title slide. And you can see here over the course of a year, this system fluctuates multiple times between a carbon source and a carbon sink. And likewise, fluxes of water are extremely variable as well. And if you're a person who works more with flux data in mesic ecosystems, that kind of represents a little bit of a contrast. Usually in these wetter systems, you'll see kind of a standard, seasonal type of curve of both of these fluxes. Now, unfortunately, this really highly variable carbon water cycling is pretty poorly captured by most models. This is a figure from an awesome paper by Natasha McBean from 2021 that compares a number of terrestrial biosphere model net ecosystem exchange with net ecosystem exchange that's measured at a number of flux tower sites. And what you can see is if you compare the variability of what's on the y-axis, which is the modeled NEE, with the variability and magnitude too of what is on the x-axis, which is measured with flux towers, they're quite different. So it seems really imperative that we improve our understanding of these highly variable fluxes, and there are a couple of ways that we can work towards those aims. First off, which has been identified in a number of papers now, including ones by Natasha McBean, is just improved representations of model hydrology and improved representations of the interactions between hydrology and vegetation physiology. But if you know me, I am not a modeler in any capacity, but I still think there's a really important role for people like me in tackling this question, which is we can supply better data sets of dryland meteorology, hydrology, and fluxes. The second kind of way we can improve our understanding of these really transient fluxes is really a better understanding of how some of these transient kind of meteorological or hydrological dynamics really impact vegetation physiology, which kind of necessitates making all of these mechanistic links between these processes across the whole soil plant atmosphere continuum. And it's with these goals in mind that we decided to kind of make all of these linkages last summer. We have an Ameriflux rapid response system tower, so shout out Ameriflux for that, in southeastern Utah, where we're measuring fluxes of carbon and water and all of these different meteorological variables. We also have a number of a nice depth profile of soil water potential from the surface all the way down to bedrock, so we can link what's going on in the soil with what's going on at the ecosystem scale. Probably the most novel aspect of what we did last summer is within the footprint of the flux tower on the dominant tree species at the site, we installed 14 different stem psychrometers, which are these really neat instruments that can measure plant water potential at the same exact time step as the flux tower measures fluxes. Every 30 minutes, these measurements are taken. So we had three main goals for this project that we did. First off is, what really makes this ecosystem and the vegetation, this ecosystem tick? What are the primary drivers of plant water potential and ecosystem fluxes? We also wanted to understand, what's kind of the linkage between plant hydraulic status and carbon and water cycling in this ecosystem. These processes are really rarely measured together, and even more rarely at the same frequency. And finally, we want to kind of take a step back and look at, some of these how transient moisture inputs, kind of the dominant influence over, the hydrology of the system really impacts ecosystem function. So the first thing that we found is, probably shouldn't surprise you given what I've presented already, is that, fluxes of carbon and water in this ecosystem were extremely variable throughout time. This is net ecosystem exchange, gross primary productivity, and evapotranspiration over the almost 3-year record now at the site. And if you look, you'll notice that there are really kind of two peaks in ecosystem fluxes at this site. We see one, kind of in the late spring, and we see one in the late summer. But you might also notice that these peaks aren't exactly consistent. They don't occur in every year, right? We didn't see this late spring peak or not as big of a late spring peak in 2021, and we didn't see this late summer peak in either 2019 or 2020. And the reason these peaks in ecosystem activity are not consistent is because the sources of moisture that drive these peaks also aren't consistent. That late spring peak in ecosystem activity is really driven by the melting of snowpack, which you can see here with this kind of middle snow depth graph. When that was disappearing and it was getting warmer, that's when fluxes of carbon and water started really going. And likewise, we see that the late summer peak in ecosystem activity is something that's driven by really kind of small transient precipitation pulses, largely driven by the North American monsoon system, which also does not happen in the site year to year all that consistently. The final thing here is I want to draw your attention to how these inputs of water impact soil water potential. On the bottom graph here, you can see that When we did have this, this snowpack that melted, that was really the only thing that was able to affect these deeper layers of soil moisture, which is kind of the, the teal lines here. And shallow soil water potential, which are these kind of lighter tan lines, were really just kind of highly variable and responded to more summertime water availability. So not only did water supply ecosystem fluxes fluctuate pretty rapidly at the system, the mean water potential of the stand via those stem psychrometers was also highly variable. These are daily averages of pre-dawn and midday leaf water potential over the course of the measurement period in 2021. And you can really see kind of this initial summer dry down, and then the monsoon comes and it wets up and then dries down again. And then we near the end of the growing season. It's also important to note here that that was the year where we didn't have much snow accumulation. And so deep soil water potential is pretty much flatlined throughout the entire year. And so because deep soil moisture was so static, you really see that stand water potential track fluctuations in shallow layers of soil moisture. And sure enough, we can also see that statistically, not just visually. We found that the kind of overwhelmingly the most important factor controlling variability in all of these ecosystem processes was shallow mid layers of soil water potential. This is a correlation plot which just shows the Pearson's R between daytime and growing season fluxes and water potential and all of these different atmospheric or soil water potential drivers. And a few things to note here. Number one is that really atmospheric drivers played very, very little role in dictating the variability of all of these processes that we measured. It really was just shallow soil moisture. But deeper kind of middle layers of soil moisture, and actually some of the deeper layers of soil moisture did matter a little bit more for controlling carbon fluxes than any sort of hydrological process, which might make sense given the rooting depth of a lot of this vegetation. So at first it was a bit puzzling as to why atmospheric drivers here really didn't matter. There's so much great work out there showing the proximal role of VPD or driving plant water potential and fluxes, and mechanistically it should be and probably is linked to carbon water dynamics. But think back to how the seasonal cycles of all of these meteorological drivers work at this site. We have these two pulses of moisture in late spring and late summer. And during those periods, the other meteorological conditions are actually quite different. During the late spring, it's much cooler, there's lower light availability. And during the late summer, when the monsoon rains come, it's really hot, it's really dry, there's high light availability. So our explanation for this is that there's just this decoupling between what's going on in the atmosphere and what's going on in the soil. So I think now we have a pretty concise answer to this first question here. And it's really these shallow to mid layers of soil moisture that are controlling ecosystem functioning. And the answer differs slightly if you're talking about fluxes of carbon versus fluxes of water and stand water potential, but I think the message is largely consistent here. I think this result also really fits nicely into some of these ongoing debates about the role of VPD versus soil moisture in dictating ecosystem function, and deep soil moisture versus shallow soil moisture. And this is just one piece of the puzzle, but I think what it indicates is that the role of all of these different factors is likely just due to how strongly these drivers are coupled and how much variability over the course of a season there is in these different processes. Okay, now on to the second question here. Is stand water potential linked to fluxes? If you're a person who is involved in any sort of plant hydraulics research, you probably already know the answer to this, and that it's yes. We found pretty solid linkages between both daily pre-dawn water potential and daily midday water potential with fluxes of carbon and water across kind of all time points, all fluxes, which really indicates something I think quite intuitive here, which is that fluxes of carbon and water during the daytime are impacted both by the mean aridity of the system, reflected by pre-dawn water potential, and also how vegetation regulates itself around that mean aridity, which would be reflective of the midday stem water potential. So again, a pretty straightforward answer here. We saw really tight linkages across the whole soil plant atmosphere continuum. I want to return again to the reason why all of these processes were so coupled together, and it's because they're all mediated by those same two sources of moisture: winter snowmelt and these transient pulses of summer rain. So the remaining question really is, how does variability in the kind of the presence or magnitude of those moisture inputs affect the ecosystem? Well, we really, only had one winter where there was a significant snowpack. So I think going into more detail on that is best left for a longer data record. But we did see a good amount of variability in summer rain conditions. And so we could kind of do some analyses looking at how much and how long these kind of pulses of summer precipitation impacted ecosystem function. So to do this, we used a slightly modified version of something called superposed epoch analysis, which is used somewhat frequently in the tree ring community. And all this is you have a time series of data, you identify precipitation events above 2 millimeters in a given day, which is a pretty small amount. And then for the month after that event, or at least until the next rainfall event, there is just a bootstrapping approach that's used to figure out if that process is stimulated following that rain event relative to its baseline variability. To show this visually, I have just scaled all of these processes that we've measured so we can put them on the same graph and then put a smoothing line through it. And the color bars at the bottom here are indicating when this analysis showed that this process was stimulated following a precipitation event. So kind of tracking the day of the precipitation event at 0 to a month after the precipitation event. What you'll see here is that even after really small pulses of precipitation, carbon fluxes were stimulated for a really long time, up to 3 weeks kind of in the aggregate. And hydrological processes were stimulated for between 1 to 2 weeks afterwards. You can see that these two shallow layers of soil moisture remained elevated for kind of 1.5 to 2 weeks following the rain event, and stand water potential was elevated for around like 5 to 6 days. So how do transient moisture inputs impact ecosystem function? Well, what we really found is that these small precipitation events impacted the ecosystem for weeks. And I think this is a pretty straightforward answer, that these moisture events do impact the ecosystem, but the kind of magnitude of the response was really surprising. Okay, so just to conclude here, this is definitely kind of like a first pass look at some of these dynamics, and there's a lot of really interesting questions to answer, but I think there are a few broader takeaways here. The first is that in the region, we've already seen nearly 40% reductions in spring snowpack in the last 50 years or so, and it's really only projected to get worse. That's one of two important sources of moisture for the system. That is dwindling quite quickly, and I think as such, the future capacity of this ecosystem to act as actually a fairly strong carbon sink is really going to be increasingly controlled by these small transient pulses of precipitation in the summer. And second, I think this links back with what I was presenting in the beginning of this talk, but I think these data really show that improving some of our model schemes of soil hydrology across different depths and understanding how vegetation interacts with those different pools of moisture could go a really long way towards better representing fluxes in dryland ecosystems. I think finally, these improvements can really be facilitated by both better observational networks of soil moisture across the region, as well as some of the new exciting ways that we can remotely sense plant water potential and soil moisture. So with that, shout out to my collaborators on this project and all of the different agencies that have funded it over the past couple of years. And as I mentioned before, I'm happy to take any questions in the chat for the next few minutes. So thanks.
Great, thank you so much, Steve. That was wonderful. Right, so if you joined us late, in general, what we're going to do is have Q&A at the end, but Steve actually has to leave. So go ahead and put your questions in the chat and he'll get to as many as he can. Thanks very much, Steve. That was awesome. Okay, so we are going to move on to our second speaker now, Dr. Tonantzin Terrazas. And she is at the Instituto de Ecología at UNAM in Mexico. And go ahead and take it away. Thank you.
Thank you, Marcy. Can you see my slides now?
Yeah.
Hi, everyone. Thank you in advance for inviting me to give this presentation about how we have used stable isotopes to partition in ET in a semi-arid ecosystem in the northwest of Mexico. And I want to thank also to the organizers of this webinar of the Year of Water, and I hope you like this presentation. And this will be the outline of the presentation, a brief introduction, background and motivation, followed by the isotopic theory used to partition in evapotranspiration, then a case of study in a semi-arid region, and finally conclusions. Mexico is a mega diverse country which is home to many types of ecosystem. It's a country with one of the most complex topographies, and it's influenced by climatological processes that occur both from in the Pacific and in the Caribbean, and those that descend from the North Pole. This map represents an important challenge when studying different ecosystem types in our country, but also the study of ecosystem processes. More than 50% of the national territory is arid or semi-arid zone, and recent studies tell us that there will be an expansion of the dry lands in the next 80 years. Here, I would like to show some of the infrastructure that we have as a country for the continuous monitoring of ecosystem fluxes using the eddy covariance technique. As you can see, most of these sites are, we call them observatories, are operating in the semi-arid and arid regions of the country, which are basically here in the north of Mexico. Semi-arid and arid regions usually receive less than 500 millimeters of rainfall per year. These regions support many vegetation compositions and structure, evergreen and deciduous shrublands, perennial and annual species, C4 and C3 grasses, and the timing and the amount of rainfall is the primary limiting resource for these ecosystems. We also know that the global drylands dominate the internal variability of the carbon sequestration, and they play a major role in the carbon cycle. In many semi-arid regions, such as the northwest of Mexico, evapotranspiration is the second largest component of the hydrological cycle after rainfall precipitation. During the wet season, in previous studies, we have found that evapotranspiration represents more than 50% of the total rainfall in a shrubland like this one in the photo. However, this flux, ET, can account for 80 to 100% of the annual precipitation in many semi-arid regions. Despite that evapotranspiration and the ratio of ET is highly variable, there is a tendency for transpiration to be the dominant flux, especially in these ecosystems that are dominated that have a dependency on water. And a recent worldwide review showed that transpiration can represent 60% of the ET in different types of ecosystem and around 45% in shrublands and deserts. So techniques to measure ET have improved significantly to address questions regarding ecosystem functioning and water balance. Among the water cycle components, ET partitioning into its components of transpiration from the vegetation and soil evaporation remains difficult and challenging. So scientists usually combine different techniques to do this partitioning. So a few models have been developed in the past year related to water use efficiency or remote sensing data, yet field validation data are needed to refine model approaches to the scale of the ecosystem. When partitioning ET, the fundamental problem is the spatial representation to separate these two fluxes, or this flux into the two components of transpiration and soil evaporation at the ecosystem scale. So combining eddy covariance with stable isotopes, it's an alternative to partitioning ET at the ecosystem scale. Stable isotopes of water can be used as tracers in the hydrological cycle because these have different fractionation processes that can be predictable with temperature and humidity. So the big assumption is that we have two sources for the mix, which in this case is evapotranspiration, and we only have two sources that can be soil evaporation and transpiration. The difference is in the isotopic composition of these two sources is what allows us to partition in ET. That being said, we have to combine the two different techniques. While the eddy covariance technique carries a direct measurement of ET over the ecosystem canopies, with isotopes, we can know the fraction of the vegetation by applying this mass balance equation in delta notation. So with this figure, I'll try to explain how do isotopes work if we have the rain that comes with an isotopic composition of -9. That water that goes into the soil eventually is taken by the plants and then transpired with the same isotopic composition. This is what we call the steady state conditions. On the other hand, which is shown in the right, in non-isotopic steady state conditions, isotope change the value depending of the water turnover of the plant. In order to apply the mass balance equation, we have to measure these three components or the isotopic composition of these three sources. First, for the isotopic composition of evaporation, soil evaporation, we can take samples of the first centimeters of the soil and then apply this model to estimate soil evaporation. And basically we add here physical components such as the relative humidity and some factors. However, for estimating the isotopic composition of the transpiration, it gets a little bit more tricky because although most isotopic-based ET studies assume transpiration in isotopic steady state and estimate delta T is equal to the source to the water source of the xylem water, delta T can be estimated in non-isotopic steady state conditions and is usually modeled or measured directly by coupling the leaf to the leaf with a chamber and then to a laser that can measure the vapor that is coming up from the plants in real time. So delta T depends or is controlled by many factors such as the turnover of the water in the plant, the stomata density, the stomata conductance, and the leaf content, and a specific species effect on the local isotopic signals that can be difficult to identify and to distinguish. And finally, to estimate the isotopic composition of evapotranspiration, we can apply this mixing model, the killing model, which basically we need to have the concentrations of the isotopic value in a concentration gradient of humidity along the canopy of the ecosystem, and then the intercept of this regression is what allows us to identify the delta ET value. So with this background, my colleagues in the northwest of Mexico and I aim to partitioning T over ET in a shrubland in the northwest of Mexico. We used the eddy covariance data and isotopes, and we collected many samples of plants, soil, and water vapor for stable isotopes analysis. And this is a case of the study. This is a shrubland, which is located in the middle of Sonora State. And basically here, and this is a seasonal ecosystem that has a dry season and a wet season, and it's controlled by the North American monsoon system that brings moisture or rainfall during the summer months from June to September. And this was our study site. So here we collected different samples, and it's very important to know that this is a multi-species shrubland. we have so many different species in the ecosystem. And we collected leaves and stem samples from these different species. And we collected soil in the first 10 centimeters of the soil, which were important to estimate delta E and delta T for transpiration. At the beginning of this experiment, we were catching water vapor in this gradient along the tower every 2 hours to catch water vapor for the isotopic analysis. And we were trapping these samples using a cryogenic liquid nitrogen. But then eventually we switched with a lot of effort and work to maintain a laser running in the field. We switched our measurements in later years to have continuous isotopic analysis of the vapor using the laser in the site. This, however, requires a lot of fieldwork logistics, and it can be very difficult to execute at the site. For the stem, soil, and cryogenic vapor tramps that we use the cryogenic distillation to extract the water, and those waters were analyzed in an isotopic analyzer to get the isotopic composition. Then we apply the equations that I showed earlier, and for the isotopic composition of the transpiration, we use 3 different approaches. We consider the isotopic steady state conditions for the plants, the non-steady state, and we propose this non-steady state, but we were weighting the relative importance of the dominant species. The species, the vegetation cover, and we were accounting the relative values of the stomata conductance. So some of the results, which we are still working on it, are shown here. We have here the different delta T of the isotopic composition and for 4 different species. And as you can see along the day, they have different isotopic values, and they are also different between the species. And this gets very tricky when we want to obtain a single value for the delta T of the ecosystem. And here we were adding the isotopic composition of the second sort, which is the delta E of evaporation, and the mix by applying the killing plot. And we can still see the delta T for each of the species that we were measuring, and we still have that challenge. So when we were applying the equation and doing some average for the species or weighting the delta T values, we found these that I'm showing in this plot. We have the 3 different approaches for estimating delta T. And you can see here how many of these values overlap, or basically they overlap. And the uncertainties of this delta T increases, especially for the one with the relative importance incorporated. So when applying the mass balance equation to partitioning the ET with the ratio of transpiration and evapotranspiration, we found a lot of uncertainties associated with this different approach, and we saw that the error propagation were increasing with a more complex approach. So in general, we found that independently of the 3 delta T methods applied, or weight to partitioning, transpiration was accounting for 30 to 100% of the evapotranspiration at the site during the wet season. So some of the conclusion of these studies, or some of the things what we have been learning, is that it gets very difficult when we have multi-species ecosystems dominating the ecosystem. And so we have different problems when we want to estimate the delta T value. And we observe large uncertainties with these values. And so these findings are important because it helps us to design new experiments. And we also know that it can be okay if we assume steady state conditions to partitioning T over AT. And for this ecosystem during the wet season, transpiration, it was the largest component. And I think, well, thanks to many institutions and colleagues are behind this work. Thank you.
Wonderful. Thank you, Tonantzin. I'm sorry. I haven't practiced ahead of time. Thank you very, very much. That was awesome. Okay, so please go ahead and put questions in the chat, but then also we're going to do a joint Q&A at the very end. So you'll have a chance to ask some questions. Okay, so let's move on now to our third speaker, Dr. Matt Dannenberg from the University of Iowa.
All right, how does that look? Excellent. All right, thank you, Russ and Marcy, for the invitation to present. I appreciate it, and I'm excited to share some work that was that I'm happy to say was just recently accepted in Global Change Biology and is now available as an accepted article there. So if you're interested in this work, I would definitely love it if you checked that out. All right, so what I'm going to talk about today is the extent to which the recent hot drought in the US Southwest during the summer and autumn of 2020 affected the gross primary production of the region, and the extent to which the loss of primary production during that drought was driven by the exceptionally high air temperatures and vapor pressure deficit that are kind of characteristic of a hot drought. Before I get into it, I want to thank all of the co-authors and funders on this. Many of the co-authors, including Marcy and Russ, are in the call here as well. So thank you all for your help and for contributing to this. So let's start by talking a little bit about the Southwest hot drought that happened in 2020. So for much of the Western US, particularly the Intermountain West, the kind of summer to autumn period is the time when most of the rainfall occurs. That's not the case along the West Coast, but at least in the Intermountain West, that's kind of the wettest time of year, especially in the Southwest. That's influenced by the North American monsoon. In the summer and autumn of 2020, specifically the July to October period, precipitation was exceptionally low compared to historic norms. So this map is showing precipitation anomalies as a percent of the normal precipitation for that time of year from the PRISM climate data record back to 1895. The baseline period for the anomalies is 1981 to 2010. And so you can see that across the region, precipitation was pretty exceptionally low, on the order of like 0 to 50% of normal for much of the Southwest. What's really remarkable, though, is that all of the places outlined in black on this map are places where that precipitation was actually a record low going back to 1895. And so something like 20% of the Western US was actually in record low precipitation territory. And so it was, for a lot of the region, the driest July to October period on record. But what was really remarkable about this is that not only was the precipitation exceptionally low, but overlaying on that were record high temperatures and vapor pressure deficit. And so now, again, these two maps are showing in the middle the maximum temperature anomalies during that period, and on the right, the maximum vapor pressure deficit anomalies during that period. And again, the black outline is showing where those were record breaking back to 1895. And so you can see that across a large percentage of the region, particularly the Southwest, we had record low precipitation combined with record high temperature and vapor pressure deficit, making this a great case study for a hot drought, a combination of low rainfall with abnormally high temperatures and vapor pressure deficit. We're particularly interested in the vapor pressure deficit side of this hot drought because vapor pressure deficit essentially represents the concentration gradient of water vapor between a saturated, evaporating, or transpiring surface and the surrounding atmosphere. And so having higher vapor pressure deficit means a couple of things. The first thing it means is that it should drive faster loss of water from both plant and soil surfaces. And the second thing it means from the plant perspective is that plants can either control their water loss by reducing stomatal conductance, or they can risk serious damage to their hydraulic function. And so higher vapor pressure deficit should mean faster loss of soil moisture, faster loss of water from the plant, and potentially lower carbon uptake as plants either lose hydraulic function or close their stomata to prevent that, that loss of water. And then further, that vapor pressure deficit, as we likely, most of us likely know, is expected to increase as temperatures continue to increase because the relationship between saturation vapor pressure and air temperature is nonlinear. And so even at a constant relative humidity, a hotter climate will have higher vapor pressure deficit. And so that means that meteorological droughts, precipitation deficits will occur or will coincide with a hotter and atmospherically drier climate. And we've seen this both in the historical record. So here, this map shows a nice analysis from Kim Novick and Darren Ficklin showing historical trends in vapor pressure deficit. And those increases in VPD are expected to continue into the foreseeable future as the climate continues to warm. And so this is from a nice paper by Ben Cook showing projections of multiple hydroclimatic variables out to the end of the century. And pretty much ubiquitously across the land surface, vapor pressure deficit is expected to continue increasing. And so that means, again, that meteorological droughts, those precipitation deficits are going to be overlain on a hotter and atmospherically drier background climate. And so what we wanted to do in this research is use the 2020 Southwest hot drought as kind of a case study or a natural experiment of sorts to assess, first of all, how much the gross primary production of the region was reduced by those exceptional conditions. And then secondly, to kind of partition out as best as we could the extent to which those gross primary production reductions during drought were amplified by the hot part of the hot drought, namely the high air temperatures and the high VPD. And so what we did is we used soil moisture active passive or SMAP satellite gross primary production estimates to kind of do a regional scale analysis. This was kind of the core of our analysis. And so we used that SMAP gross primary production product to estimate the total loss of primary production across the region. And then we used SMAP root zone soil moisture along with grid met air temperature, radiation, and vapor pressure deficit to kind of figure out how much of that gross primary production change was due to these different meteorological and hydrological drivers. A fundamental issue with using a product like the SMAP gross primary production for this kind of analysis is that that's a light use efficiency model that has response functions already built in to soil moisture and VPD and temperature. And so essentially the model assumes, the SMAP gross primary production model assumes that if you have high soil moisture, you have high gross primary production. If you have high VPD, you have less primary production. So there's these kind of a priori assumptions built into that model for what the response of gross primary production should look like to those variables. And so we also used some data that's independent of any of those meteorological inputs to kind of confirm and triangulate estimates of how much these meteorological and hydrological drivers affected gross primary production. The first one that we used at the regional scale is the contiguous solar induced fluorescence product. Solar induced fluorescence basically represents the light emitted by chlorophyll and it's more or less linearly related to gross primary production, though there's a lot of caveats to that relationship, and there's a lot of work surrounding the connections between solar-induced fluorescence and GPP. But at minimum, we know that SIF is related to GPP, and it's independent of these meteorological inputs. It's a satellite-only product. And then the other thing we used was gross primary production estimates from 9 Ameriflux towers across the region, shown in this inset map, including 4 towers in New Mexico, 4 in southeastern Arizona, and 1 in California. And for that analysis, we used gross primary production as the response variable, and then root zone soil moisture, temperature, and radiation and VPD estimates from the tower as well. To answer our 2 questions, we basically followed a 3-step approach. The first thing we did is we just simply calculated the total gross primary production anomaly in both the SMAP record and at the tower level relative to a 2015 to 2019 baseline. That's a pretty short baseline that's constrained by the period of record for SMAP, which was launched in late March 2015. So unfortunately, it's not a particularly long record from that SMAP satellite. And so that kind of constrains the baseline period. Next, we fitted an empirical model that relates monthly gross primary production anomalies to monthly anomalies in photosynthetically active radiation, soil moisture, air temperature, and VPD. We attempted as best we could to account for the fact that all of those meteorological drivers are intercorrelated. But then we basically used that model to then decompose the observed anomaly into its constituent parts. And we did that by taking that fitted empirical model and basically running it with 1 driver at a time to try to estimate the extent to which the gross primary production anomaly was driven by each of those meteorological and hydrological drivers. And so I'm going to show a few figures that look sort of like this one on the left, which shows what the model thinks the gross primary production anomaly should have been during July to October 2020. And then any difference between that bar and this white line indicates unresolved variance in that simple empirical model that we have. And then all of these colored bars show the components of that gross primary production anomaly that were, according to the model, attributable to PAR, soil moisture, air temperature, and VPD, going from left to right. So in total, during the 2020 drought in the Southwest, the region lost more than 120 teragrams of carbon compared to the 2015 to 2019 mean for that time period. So this time series is showing the SMAP observed or SMAP estimated gross primary production in total across the Southwest from 2015 to 2020. Again, 2015 is the first year available from SMAP. And you can see that prior to 2020, gross primary production averaged somewhere around 450 teragrams over that July to October period. But in 2020, it dipped to more than 25% below that amount. And so it was a loss of something like 120 teragrams of carbon. On the scale of the global carbon cycle, that's not a huge amount, but for the Western US, that's pretty significant, especially when we start to think about the kinds of uses that land is put to. So for example, a lot of the land in the study region is devoted to ranching, and the amount of gross primary production that was lost during that drought is roughly the amount that it would take to feed 45 million head of cattle for a month. So it's a pretty substantial loss of primary production. And even that amount, that 120 teragram amount, is probably underestimated. This bar graph is showing a comparison between what SMAP thinks the loss of primary production was to what the 9 Ameriflux towers say the anomaly should have been. And you can see that in most cases, SMAP significantly underestimates the magnitude of that anomaly. Particularly at the southeastern Arizona sites, which are the ones on the left in this bar plot, where the effects of that drought were most strongly felt. And so what this suggests, I think, is partly that, you know, as Steve was saying earlier, we need to improve the way that models represent carbon and water fluxes in drylands. A lot of other folks, including some in this call, Joel Biederman, Mallory Barnes, and others, have shown that it's pretty common for remotely sensed models to underestimate the magnitude of gross primary production variability in dryland systems. And so I think it's pretty important that we do a better job of capturing that. Across the region, we see pretty much ubiquitous losses of primary production. This map is showing the gross primary production anomalies through space across the study region. And every one of the 6 ecoregions that we looked at had pretty substantially reduced primary production compared to their 2015 to 2019 means. And in most cases, it was well below the normal range of variability for those systems. I'm going to highlight 2 in particular, the semi-arid prairies, which are here in the eastern part of the study area, and the Sierra Madre Piedmont, which is southeastern Arizona, where we had particularly large losses of primary production during this drought of roughly 40%. In the Sierra Madre Piedmont, not quite as extreme in relative terms in the semi-arid prairies, but still roughly 100 grams of carbon per square meter that was reduced by that drought. Again, though, at least in southeastern Arizona, that's very likely underestimated. This figure is now showing the same gross primary production anomalies for the 4 southeastern Arizona flux towers. And you can see that while SMAP says the overall loss of primary production in that region was something like 40% of the mean, at these 4 flux towers, it's something that's more like 70 to 90% of the mean that was reduced by the drought. So it was a pretty exceptional reduction in primary production. According to the attribution model that we ran, most of that primary production loss came from reduced soil moisture, a little bit more than 50%. So this figure is again showing the decomposition of that gross primary production anomaly into its constituent parts. And just over 50% of that gross primary production anomaly was predicted to have come specifically from low soil moisture. But that was pretty substantially amplified by high vapor pressure deficit. About 40%, a little bit less of that gross primary production anomaly came specifically from high VPD. That's pretty consistent with what we also saw at each of the towers individually. Using the flux tower data, the magnitude of the soil moisture anomaly is a little bit higher than SMAP thinks it was, and the magnitude of the VPD anomaly is a little bit lower than SMAP thought it was, but overall still fairly consistent, where roughly 40 to 70% of the gross primary production reduction was coming from low soil moisture, and roughly 30 to 40% was coming from VPD. Again, like I said, the VPD component was maybe slightly overestimated by SMAP, likely due to the fact that it includes that a priori assumption about how gross primary production will respond to high VPD. So that's likely accounting for that's likely the main reason that SMAP is slightly overestimating that. However, in conclusion, what we found is that during that 2020 hot drought in the Southwest, gross primary production was reduced by at least 25%, probably more than that, considering how much SMAP underestimates the tower observed loss of primary production. And most of that primary production loss was coming from low soil moisture, but it was pretty substantially amplified by the high vapor pressure deficit, and to some extent by the high air temperatures that are characteristic of a hot drought. And with that, I will close. Thank you.
Great. Thank you so much, Matt. That was awesome. Okay, so let's move on to our final speaker, last but not least, Dr. Kiona Ogle, who is at Northern Arizona University in Flagstaff.
Thank you, Marcy. Let me just get this set up here. I don't know where everything went. Okay. All right, here we go. Yes, first I just want to thank Marcy and the organizers for inviting me to participate in this webinar. And it's really great to see how many people are online for the webinar. And I'm going to start out this talk today with actually an Ignite talk I gave at ESA a few years ago to help set the stage for this webinar talk, where I aim to highlight questions and approaches related to understanding timescales over which indices of moisture availability influence carbon and water fluxes. And along with collaborators, my lab group has recently been leveraging the rich information contained in flux tower data to address these questions in the context of understanding drivers of ecosystem carbon-water exchange. And my intention here is to give a brief overview on potential approaches that may be of interest to this group. So as an overarching question that plant and ecosystem ecologists are often interested in is what governs plant and ecosystem functioning. And I think we generally agree that exogenous factors such as those related to soils, climate, and weather, and endogenous factors representing internal plant or ecosystem feedbacks are important drivers of plant and ecosystem behavior. For example, in the semi-arid ecosystems that my lab studies, moisture availability is often one of the most important drivers of plant and ecosystem functioning, but the timescales over which moisture dynamics and other drivers affect plant and ecosystem processes is not entirely clear. The typical approach, though, to evaluating drivers of ecosystem functioning is to relate observed variables such as carbon or water flux components to concurrent drivers such as soil moisture status at the time of that measured response. However, it's more likely that concurrent and antecedent conditions simultaneously govern plant and ecosystem functioning. Furthermore, past conditions may be even more important than current conditions, as represented by the thicknesses of these arrows in this figure. But how do we know the timescales over which such drivers influence the system? That is, which of these arrows are the thickest? It is even more likely that antecedent and concurrent exogenous and endogenous drivers interact to govern plant and ecosystem functioning, but their timescales of influence could differ greatly. Some may operate on the order of hours, while others may operate on the order of weeks or longer. So how do we evaluate the importance of past conditions? How do we determine the timescales over which various driving variables influence our processes of interest? This may be achieved by collecting data or designing experiments to cover a wide range of conditions that produce varied combinations of concurrent and antecedent conditions and that generate response and driver time series. And long-term flux tower sites provide exactly these kind of data. And combining those data with modeling approaches will allow us to uncover these timescales of influence. I want to walk through a toy example to illustrate this timescale issue. So this is based upon fake data that just simulated. And we have a response variable here, Y, and a potential covariate X that's been measured over time. So maybe this time step is over daily periods. And a typical approach, again, might be to relate Y versus X concurrently. So just take the pairs of point and plot them in the scatter plot. And you can fit a line in this toy example. You can see that this is a positive but very weak relationship. But what might actually be happening in the system is that it's not just concurrent values of the covariate, but past conditions are potentially just as important or more important. So to evaluate the importance of past conditions, you can think of taking like a weighted average of X over time. And so here's just 3 hypothetical examples of how you might do that weighted average. This first graph here in B1 shows the importance weight that we might use for that weighted averaging for values of X measured concurrently 1, 2, 3, 4 days ago. And the height of the bars are all equal, indicating that this would just be simply calculating a numerical average of 5 values of X, including the current value of X. Plot B indicates importance weights that put a lot of importance on the concurrent value and the importance of past conditions decay over time, whereas plot B shows the potential for a lagged response, such that conditions 2 to 3 days ago are most important potentially. So if we take those 3 different examples of how to calculate a weighted average of X to get an antecedent value of X representing that system, we plot the observed value of Y versus the antecedent value. In this example with B1 equal weights, just to average, we get a negative relationship, albeit still pretty weak. If we use a second set of weights that give a lot of importance to concurrent values, we still get a positive relationship like in D0, but potentially it looks a little tighter. If you take this last example that assumes a 2 to 3 day lagged response, we get a negative relationship and it's exceptionally strong, so very tight, high R squared. So if I were to pick which of these scenarios likely explains how X it drives Y, I would pick this plot in D3 and conclude that there's likely a 2 to 3 day lagged response between X and Y. And so one of the goals of a lot of the work in my lab group is to understand or estimate these importance weights and the associated antecedent effects as the strength of that relationship and the direction of it. And the approach that we have been using is called a stochastic antecedent modeling approach. And I'm just going to walk through a graphical representation of that before I go into a case study. And here, each of these circles and squares represent a node or quantity in the model or the analysis, where the blue all represent data that we have collected. Y here is a response variable of interest, such as a flux variable. X, Z, and E represent potential driving variables, where X and Z are likely to represent exogenous variables. And we're going to relate Y to a predicted value that's going to be a function of the antecedent conditions, both exogenous and endogenous values. So here we'd start out with the data model that describes the variability in our observed response. Some flux variable, for example, given appropriate distribution that describes the variability in Y, given the expected value or predicted value, and sigma squared represents some sort of observation or measurement uncertainty. And then next we would describe a model. You think of this as a process model that describes the expected or predicted value of our response. It's a function of, let's say, some effects parameters and our covariates. And here I've shown the covariates as antecedent covariates, and potentially concurrent covariates are incorporated for those covariates where you know that that's X, for example, Y and Z are coupled concurrently, and antecedent coupling doesn't make sense. And within the SAM framework, the antecedent variable is calculated again as a weighted average of past observed variables, where J is a lag period going into the past. And if J equals 0, that would indicate that you're incorporating concurrent values of X. And as you go further back, you're incorporating older and older values of X. And the W here is really important. That's the antecedent importance weight. We assume that we don't know that a priori, and we're going to estimate it by fitting this model to our actual data that we have to infer those importance weights. And we often implement this model. It actually often looks like a regression type model. We implement it frequently within a Bayesian framework, where we give prior distributions for the parameters in model, including importance weights, any variance or uncertainty terms, and alpha here would represent any sort of sensitivity parameters or effects parameters. Importantly, these importance weights can be thought of as like probability parameters. It's a probability that conditions 5 days ago continue to influence your response that you observe now. So those W's must sum to 1 over those time steps, and they have to each be between 0 and 1. So that Dirichlet distribution is just one mechanism for obeying those sort of mass balance constraints. And then within the framework, we might often allow those effects parameters, those coefficients, to be modeled hierarchically to represent aspects of the sampling design, such as plots nested within site or individuals nested within species, for example. And then we have this model that we fit to the data on our responses and our covariates. And we've applied that kind of model to a variety of data and processes spanning a wide range of temporal and spatial scales. But today I'm just going to focus on applications directly related to this webinar, and including data collected from flux tower sites related to NEE and ET, and to some extent also soil and ecosystem respiration. And I want to use this study here published by Yao Liu, a former postdoc in my group, to illustrate the application of SAM and example inferences that can be drawn from such application. And here we focus on daily NEE data from the global FluxNet network, spanning about 50 sites and over 190,000 site days. And you can see, different sites have different numbers, different amounts of data depending upon the temporal depth of that data based upon the size of that circle. We have all these data then that we fit a SAM type model here, and I'll walk through the model structure associated with this data. We have NEE data, and we are modeling NEE. Thinking of it as taking on different values or responses depending on whether it's in the growing season or the non-growing season. And in this example, we use remotely sensed NDVI data to essentially govern when the system is switching between growing season and non-growing season. And then those predicted NEE of growing season and non-growing season are expressed as functions, like in a mixed effects kind of linear regression model. There are functions of concurrent soil water at the time of that measurement, but also antecedent conditions with respect to radiation, temperature, vapor pressure deficit, soil water content, and precipitation that was recorded at each of those sites. And we also incorporate an autoregressive effect for NEE. So this represents the NEE that was measured yesterday to account for some of an endogenous effect within the system. These antecedent variables of radiation, temperature, VPD, and soil water contents are calculated over relatively short time scales that represent weighted averages occurring up to potentially 2 weeks with weights that are at the scale of daily to weekly weights. And precipitation here is incorporated over a much longer time scale to represent the influence of precipitation beyond what was recorded in relatively shallow or moderate depth soil water content sensors. So large precipitation events in the winter may not actually have been represented in the soil water sensors at these sites. So we have these longer scale precipitation factors in here. So this is all applied within that SAM framework and fitting those models to 50 some sites. I'm just going to give you an overview of the results broken down by the biome from which those sites are aligned with. And we use this model to evaluate the relative importance of past conditions, which we describe as a memory response. So environmental and biological memory, or just combine them and think of this as ecological memory, the role of antecedent conditions. The R square basically tells you how well the model fit with, let's say, only concurrent values incorporated. That's the yellow and light blue. And then once we add the antecedent values, we improve the R squared according to the dark blue and the dark green. And so the takeaway here is that this ecological memory, or the role of antecedent conditions, explains an additional 1/3 of the variation in NEE in drylands, especially grasslands and shrublands. And in these systems, antecedent conditions are just as important or even potentially more important than concurrent conditions in terms of moisture indices. We can also look at how the importance of those antecedent conditions, as described by that ecological or environmental memory, it aligns very well along an aridity gradient. So we have sites that are plotted from relatively wet sites to drier sites as you move to the right on this plot. As you go into drier sites, the importance of those antecedent conditions or that environmental memory is stronger in those systems. And globally, the importance of antecedent drivers scales with aridity, both within a particular biome. So for example, across the grasslands, but also overall across all the biomes that were explored. And I'm going to highlight a second one that focused specifically on arid and semi-arid systems, since that analysis of over 50, flux tower sites suggests that these arid and semi-arid systems are really governed by antecedent conditions and have a strong environmental memory. We then conducted analyses at the New Mexico elevation gradient network that Marcy runs to explore this in more detail at these arid systems. And I'm just going to show some results from the importance weights related to drivers of vapor pressure deficits, air temperature, precipitation, shallow soil moisture, and deep soil moisture. And this is done for 2 different sites, woodland sites in this system for evapotranspiration, that's the left column, and NEE, the right column. And there are a few things I want to point out. So each of these symbols represents a relative importance of, say, deep soil moisture at different time steps into the past. And if we just focus on the moisture drivers, we can see that concurrent or shallow soil moisture, that's the grayish symbol here, has relatively high importance concurrently. So there's a very short memory. Shallow soil moisture is most important on the same day or nearly the same day. But if you go to deeper soil moisture, the dark filled symbols, the relative importance increases as you go further back in time. And there's, in some cases, especially at ponderosa pine, there's up to a week lag between deep soil moisture and ET and NEE. And then if you look at the role of precipitation, antecedent precipitation, you can see that some of the importance weights are pretty high as you go further back into time. So 2, 3, 4 months ago represents sort of like the past season. So precipitation is really important at much longer time scales. And what's interesting here is that all of these moisture-related drivers or indices are interacting across these timescales to affect NEE and ET. So we find very strong interactions among these factors within their analysis. And these interactions among factors are really interesting because across many different studies and flux data, we find that their ecological response, whatever flux variable, is clearly driven by concurrent conditions such as moisture and temperature. It's also clearly driven by antecedent conditions, but time scales over which those operate is not consistent across sites necessarily. But the thing that we often always find is that the antecedent conditions appear to moderate the response to the concurrent values. So here's just a quick example to illustrate that with respect to the moisture interaction of antecedent and concurrent moisture. So for example, if you look at, if it's been fairly wet for several days in the past, as might occur during the monsoon season in the Southwest, and if you add additional water in the form of rains, we observe little to no response in our flux once you add that water. But if you look at a scenario where it's been dry in the past, such as pre-monsoon, and you add the same amount of water, you see a huge response. So this indicates that plant and ecosystem responses to moisture inputs are contingent upon moisture history, right? And that interaction term is really critical or important. And we find that across a variety of flux components. The same kind of thing we find for temperature. There's a strong negative interaction between antecedent temperature and concurrent temperature. And the takeaway here is that basically that represents a temperature acclimation response that we see across a variety of flux components. And which I won't go through given the time, but I just want to summarize here that antecedent and concurrent moisture indices govern ecosystem fluxes. The timescales of influence are not known a priori. We have to estimate them from data and models. And the SAM approach can be used to evaluate those timescales and their effects. But I do want to point out that there are other methodologies that can give similar types of insights, such as machine learning methods, and even that superimposed epoch analysis that Steve talked about at the beginning of the webinar. So the SAM is not the only way to do that. But I think what's interesting is that SAM can allow us to infer specific timescales of influence quantitatively to infer lags, and also to make inferences about sensitivities to drivers, that is those specific effects of moisture indices. And I will just leave it there in the interest of time. And thank you very much, Marcy, for moderating the session.
Awesome. Thank you, Kiona. Well, maybe for all four speakers, let's give them a virtual or a real round of applause. Really appreciate it. Let's see. So we have about 15 minutes for questions. So Russ, I'm going to ask you to help me moderate now so that we can get everybody. And Christine, can people just unmute themselves and ask a question?
Yes, they should be able to.
Oh, great. Okay. Go ahead, Chris.
Is that me? Oh, hi. Thanks for great talks. Kiona, I had a question about, I assume that process models do a terrible job at capturing antecedent effects, but have you analyzed process model outputs of fluxes to see how they do and ways in which we can improve those models to get these antecedent conditions correct?
Yeah, I mean, I haven't done that directly myself. I have collaborated with people, both Bill Anderegg and Christopher Schwalm, and I can't remember her name, another person published a couple of papers about that and showed that like large scale biospheric models are not capable of capturing legacy effects, which is not necessarily the same as these antecedent effects. And I mean, it could be because they lack specific processes related to temperature acclimation. I know that people have written about that's not well represented in some of these process-based models. And it could also be that, you know, they don't represent labile carbon storage and how that affects response to past conditions or changes in rooting distributions or up or down regulation of root behavior. So it's like, I'm sure there's like a bunch of potential mechanisms, but in general, yeah, larger scale process models generally don't capture that memory or those legacy effects very well.
Okay, thank you.
I think we have an earlier question for Tonantzin. How do you, I don't know if you saw this or answered it. I didn't see your answer in here, but a question about how you get delta ET and how you deal with the differences in the temporal resolution among delta E and delta T.
Yes, I saw it. I was actually chatting with Xiangming. And I was mentioning that for when we took the laser to decide, we were able to take our measurements every minute, and we were averaging these values to match the isotopic composition with the same resolution of the eddy covariance measurements, and same for the E, delta E. So we got so many samples.
Great. Russ, you have a question?
Yeah, I have a question for Kiona. Can you hear me?
Yes.
So I was wondering, like, if you could try to tie the timescales that you're seeing to like a response of the ecosystem that you could somehow measure, like say it's ecosystem structure based on LAI or something like that. If you could take the time series of LAI, say from MODIS from the same sites, and then see what how the response, if you get a similar kind of time scale of response or something that you would allow you to tie it to the ecosystem structural changes, that might be the reason for the memory.
I mean, that could be one factor for possibly as it changes in ecosystem structure.
Or maybe
for
it's
the fluxes.
percent cover or something like that.
Yeah, I mean, in one study, this was actually a long time ago that Greg Baron Gafford did with me also, he analyzed soil respiration data using this kind of framework. And we found it interesting, like 4-day lag or something between respiration and I think it was vapor pressure deficit. And that 4 days matched really well what you expect for like xylem transport times. So, I mean, there could be things that you could measure with labeling techniques, or maybe is associated with vegetation structure that you could couple with other data sources. But yeah, I think there's a variety of potential mechanisms, and it would be great to be able to measure them in the field.
That may be like using say at a grassland site where the changes in structure would be more measurable, like percent cover of your dominant vegetation, dominant grass.
Yeah, and I could see that being useful for trying to understand the longer lagged effects associated with deep soil moisture and precipitation for sure. Maybe not these like 1 to 2 to 4 day lags associated with shallow soil moisture or vapor pressure deficit. Those might be more related to physiological mechanisms.
Yeah, thanks.
Kiona, I'm not sure if you saw this question in the chat from Yijian Zhang. It's very interesting to see how SAM can be applied to understand ecosystem functioning across multiple timescales and drivers. I'm wondering if the machine learning or deep learning types of algorithms could be applied here to do similar tasks as SAM. Or is
so that was like my last slide, which I went through very quickly. I haven't done it myself, but there, Marcus Reichstein's group, which there are those 2 papers I briefly showed by Kraft et al. And Besnard et al. They applied long-term, short-term memory data machine learning methods to quantify memory. I think it's definitely a potential avenue for doing that. What I think, so one of the benefits is probably it's computationally faster to do than some of these Bayesian frameworks, but the potential drawback is that you don't gain as much actual inference about, the sensitivities of the system to different drivers and so forth. So there's kind of, it just depends upon your goal, but certainly there's a potential role for machine learning methods to lend insight into these memory responses.
great. Any other questions for any of our speakers? Looks like Matt, you answered a few in the chat earlier. Yes, John, they were talking about that earlier, Dannenberg, Cannenberg collaboration. I think it has to happen. All right, last chance. Anybody have any other questions? All right, well, let's thank all the speakers again and really appreciate you guys putting together these great talks. It was really, really interesting.
Thanks for having us.
Yeah, thank you.
All right, well, thanks everybody for tuning in, and hope to see y'all in person at some meeting in the near future. All right, bye everyone. Thank